Good morning, everyone. Thank you so much for coming. And thank you to the organizers for inviting me to this amazing place to talk about my research. My name is Dr. Uljana Hesse. I work at the University of the Western Cape, where I have established the Rooibos Genomics program. So let's dive right into it. Why sequence a plant genome in the first place? Actually, knowledge on the genomic information of plants is an economic commodity. We use this in agriculture when we want to develop molecular markers which permit to select plants that have specific characteristics such as stress tolerance or production of specific biochemical compounds that are of interest to the pharmacy or the medicinal compounds. So when we know the molecular markers, when we know the genes that are associated with it, we can select these plants at a very small age. We don't need to wait for them to grow. That's molecular markers. Similar markers can be used to improve trademark protection, where this is specifically used in China, where they are used to prove the purity of the plant material in medicinal plant samples and the origin of them. But of increasing importance is actually the industrial application of the genomic information. So there is a worldwide movement to develop laboratorial processes where you identify the genes that are associated with the production of relevant compounds, and you can clone these genes into surrogate vectors, for example, into yeast or tobacco, which is easy to grow. And then you can produce these compounds independent of the actual plant, which frees you up because agricultural production of plant material is dependent on the weather, on the soil, on a lot of factors. So this is the reason why worldwide there is now an increasing movement towards sequencing plant genomes. Until recently, because the plant genomes are very big, until recently, this was very prohibitively expensive, but with the development of new sequencing technologies, this has become accessible. And if you look at this, this is a map that was published in 2021, which shows you where most of the genome sequencing is happening. You will realize that it's mostly happening in the global north, in the United States, in Europe, in China. Close to nothing is being done in Africa. When I say close to nothing is being done in Africa, that doesn't mean that the African plants are not gaining any attention. There is sequencing data out there, but the data is being analyzed in the global north. At this point, we provide the samples, but the analysis is done in the north, and this has direct implications on who owns the data. Which brings me to another point, which is specific to South Africa. So yesterday, there was already a talk about the Cape Floristic region. The Cape Floristic region is also known as the sixth plant kingdom. It is located pretty much only in South Africa, and while it covers only 0.5% of the African territory, it harbors 20% of the plant species. In total, we know of 9,000 plant species that are at home in this area, and 70% of these species are endemic, which means that they only grow in this country. Now, another very important point. When we talk about the medicinal plants of South Africa, 70% of the population in South Africa depends and actively uses medicinal plants for treatment. And over 3,000 plant species are known. Now combine these two things and you will realize that from a biological perspective, we have a biological resource that is yet absolutely untapped, because for most plant species, there is no agricultural production, and the access to these plants is close to nothing. No, not with the silicon project. That you will have to ask the PI. But I know that there was a lot of discussions when it came to getting the data back and being allowed to do the analysis. If you see now, you will see that we are part of the analysis. We have published papers, but genome projects, in particular the coelacanth, which is an intermediate between fish and land animals. So it's a shoal project, and it's beautiful biology. It's fantastic. It's actually, I never was bothered about fish until I realized how cool this one is. He walks. Like most fish, you know, they do this one. The coelacanth actually is like the mammals. Like when we walk, we move the hands like that. So when he swims like that. Originally, they thought that he jumps. I still have time. But he actually swims very slowly in a very peaceful manner, and so our analysis actually indicate that he might be the first fish that can taste sweet. And that it has, fish do not really need to have a lot of taste receptors. Later about that, I'm talking, sorry. So, that tells you why we need to do plant genomics. We have the map, we have told you about this. Now, the aim of the Rooibos Genome Project that I started in 2016 was to establish locally all methods to do plant genome research here in South Africa. Then obviously to generate a very high quality genome for Rooibos. And then because for Rooibos, this is one of the endemic plants which has an agricultural production system, so we have people that are interested in the results. My aim is to identify so the genes that are involved in medicinal compound production, desirable morphological traits, and improved stress tolerance. Okay, what is Rooibos? Rooibos is one of the few endemic South African plants that has reached international acclaim. Most people know it as a tea. It grows in a very small area in Africa, which is the Cederberg Mountain region. And it represents an important cash crop there because it's very hot in summer, 45 degrees. Is normal in February, and not many plants can survive that. So for the local farmers, this is a very important income plant. Many people know it as a tea, but not many people know just how healthy it is. It actually has been used already by the indigenous people from this region, by the Khoikhoi and the San, to treat diverse ailments, and now we have scientifically verified medicinal effects such as anti-inflammatory, anti-diabetic, cardioprotective, anti-allergic, and anti-aging effects which have been proven by scientific research. These effects are associated with the production of phenolic compounds, and Rooibos produces so for example, two of them are PPAC and asparagine, so PPAC is has only been found in Rooibos, and asparagine has only been found in two plant species, Rooibos and another plant species in New Zealand, of all the places. So these ones are actually now considered potential phytopharmaceuticals because they have anti-diabetic and cardioprotective effects. Here is one of the important slides that I want to show you. In 2009, Nestle applied for 5 patents to use rooibos and honeybush, which is also an endemic South African plant, for treatment of hair and skin conditions and as an anti-inflammatory agent. This gave an outcry because not only is this indigenous knowledge of South Africa, this is from an indigenous plant of South Africa. So it took 10 years, but in 10 years, South Africa has finally signed a landmark legislature which secures access and benefit sharing agreement between the rooibos industry and the traditional knowledge holder. In this case, they are identified as the Khoikhoi and the San. This is only for one plant species. We have 3,000. Okay? We need to protect this information and this biological resource. Okay, now before I start into the genomics program, just quickly to recap the terminology. A genome is the DNA molecules that make up the complete set of the chromosomes of a given organism. So let's start here. No, that's not. There. The plant consists of cells which contain the chromosomes, and these chromosomes are made of the double helix, the DNA. When I'm talking to a computer scientist, I would say these are 2 very long strings of letters, A's, G's, T's, and C's, which correspond to the base pairs. So adenine, thymine, cytosine, and guanine are bases of the DNA. They come in pairs, that's why we talk about base pairs. TA is paired up and GC is always paired up. So what we want to do is read this endless long string. Why I'm saying endless is because if you put the 9 chromosomes of rooibos together, you come to a total of 1.2 billion letters. If you put the entire genomic information of the human together, you get to 3 billions, so we're all billionaires. Multiple times so. Okay, I hope that this helps you to get an idea about the background. Now, what is a genomics program? The first thing that you have to do, you have to collect your samples. And then very often comes a biochemical screening approach where you need to figure out which is, of the many different plants that you have collected, which one is the most interesting one. Then you obviously want to know the genome size because you need to know when is this going to end. And then we have 2 ways of analyzing the genomic information of organisms. Either it's transcriptomics or genomics. Let's start with the genomics first. So we want to know the sequences, the A, G, C's, and T's from one of these 2 strands. But not the entire strand is informative, at least in terms of, well, that's debatable, but what the first important information that we are looking for is we want to know what proteins are encoded on the genome, because the proteins are really the tools that make our biological cells. So we will be looking for genes, which are locations on the genomes that encode proteins. The way it's done is when we want to go from genome to protein, so the genome is the same as all the cells of an organism, but they are not all the genes are switched on. Makes sense because we have skin cells, we have brain cells, we have muscle cells. You don't need to switch on all the instructions in the entire body. So the ones that are switched on in a specific tissue or at a specific time point, they produce copies of the genome called messenger RNA, And that later on is translated into a protein sequence. So when we talk about genomics, I talk about this entire genome molecule. When I talk about transcriptomics, I want to harvest the copies of those locations on the genome that have been switched on and that produce proteins. So we get information of what is actually being produced in that cell. So far, so good? Okay. So here is how the bioinformatics is being done. We go from DNA, which looks like this. So this is a small tube. Through a sequencing machine. Here is an example of the Illumina sequencer that generates, and here is the data, A's, G's, C's, and T's. These are strings. Now, you cannot generate to date the entire 1.2 billion string at one go. What Illumina sequencing does, it generates billions of 50 to 300 letter-long strings. And that is our raw data. That is what we use to recreate that 1.2 genome. Let's look what it looks like. So when we talk about genome assembly, that's exactly what we want to do. So here is this cloud of short strings, and then we use a computational process called assembling in order to regenerate your genome. And it's a little bit like trying to get the original copy, at least one of the copies of the New York Times that your cat has been playing with. So it's just to give you an idea what kind of a computational process this is. Now, once you have that, then you do the genome annotation, which refers to identification of those locations on the genome that would eventually produce proteins. So it's the transcriptome, which then, these are proteins, these are three-dimensional protein structures. It's just plain pretty. Okay, so we started off in 2016 at the beginning with Illumina sequencing, and now I understand how tiny these data sets are in comparison to what I've seen here from other people. But still, we generated 2 terabytes of raw data, which was big enough. And in total, we got 10 data sets from the plant DNA and 2 data sets from the same plant of a transcriptome, but then we generated another 25 data sets, and they were between 4 and 120 gigs per data set. Now the problem is when these reads are so short, like remember, between 50 and 300 letters, when you need to recreate 1.2 billion letter string, the regions that are difficult to reassemble will not assemble. So what needs to be done is you need to bridge that. And there is a latest technology called Oxford Nanopore Technologies, and we are using that increasingly. Yesterday it was introduced, which can generate read lengths of up to 1 million base pairs. Our longest one was 180,000 nucleotides or bases. So we improved our genome assembly. So the current robust genome is present in about 30,000 pieces, but we have, these 30,000 pieces have pretty much all the genes that need to be, so we can predict that on these 30,000 pieces, we have pretty much the entire genome covered. We come to storage. 2 terabytes is easy to store. If you look at it, the data sets are much bigger. So our data sets now for this new technology is between 10 and 600 gigs. And currently, thanks to Diplomics, we're in the process of sequencing 48 data sets. So the data is just expanding. And because I have to make sure that we don't lose the data, we need to store it in triplicates. So currently I'm working with lacy disks, but I will be talking to Derisa soon enough. So at the moment we have been quiet about everything because it's a lot of work. You can't do everything. So, but now that I know about Derisa, I want to see how we can store the data better. But currently we have it on the lacy disks, which can store between 28 and 32 terabytes. Okay. Now let's get to CHPC. Let me tell you that without CHPC, this project would not have been possible because it requires computational power to put these small strings back together. So here are steps that you have to do when you have a genome program. You need to translate, so the long reads, they come in a format, not in the letter format, they come as signals in binary files. This needs to be translated. This is done using GAPI, for which we use the Big Mem node, and we require 18 CPUs, 2 GPUs, and at least about 50 gigs of RAM in order to analyze one data set, and it will take about 12 hours. Then comes the quality filtering of all these reads, which is relatively inexpensive, but assembly, as you can imagine, between 56 CPUs and 112, depending on which program you use, between a couple of days and months. Like this one Kanu ran 3 months. And obviously it needed huge, it had a huge requirement of RAM. Like for me, this is a lot. And then, so what these assemblies do, they first put all the long reads together, but the long reads are the quality is not very good. So what you then do is you polish it with the short reads. Doesn't mean you do this one. You put the short reads on top of the long reads, you map them back, and then you identify where there are mistakes. So that's called polishing, and for this, again, you need at least 56 CPUs and substantial amount of memories. Here is, now comes the annotation. The first thing that you want to do is masking repeats. So in plant genomes, 90%, up to 90% of the genome can be just repeated elements. ATG, 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 long stretches of it. Rye, for example, 90%. So these repeats must be masked before you start looking for the information for the genes that encode the proteins, and that's done using a program called Repeat Masker. Again, lots of CPUs, lots of gigs, and substantial amount of time. Then we map the reads from the transcriptome back to the genome. So here is a genome contig. These are the reads, and you already can see the structure of the genes here. And then finally, we use Augustus, which is a gene prediction program. So on one hand, it uses the information from these reads to find the locations on the genome and identify the gene structure, but it will also use, so it's a machine learning algorithm which uses this information to predict genes in other locations based on the training data set. And this is some of our results. So Yamkela Mgwatyu is my PhD student. She will finish up this year. We have in total, we have identified 53,000 proteins or genes, of which nearly all of them, so 50,000 have been, we could identify the function of these proteins. And this graphic here, just don't look at the numbers and just trust me when I say these are the housekeeping processes. For example, energy metabolism, carbohydrate metabolism, lipid metabolism, amino acid metabolism. These are pathways, and these pathways have been identified, so we have pretty much identified the genes in all these housekeeping pathways that are responsible for keeping rooibos going. Until now, we have been only publishing methods papers. We have, so Emily led by Emily Stander and Yamkela Mgwatyu. I have done some bioinformatics work. But we are finishing the rooibos genome next year, so keep your eyes on the horizon for our genome paper. And with that, I would like to thank my co-PIs, in particular Peter, who is not here at the moment, but he was an invaluable comrade during this time. Then all the rooibos farmers that participated, as I said, we are doing it from scratch. We go to the farmers, I know them personally, we talk to them, we explain the project, and people that do not know how to read and write understand what we are about. We have the support of the local communities. Then these are people that have helped, so intermediately throughout the entire project. These are places, so these are institutions that have helped with the work, and these are funding agencies, including Diplomics. And this is my team. Yes, and with that, I'm happy to take questions.
Thank you. Thank you for a nice talk. I'll just take one burning question quickly before we move over to the next speaker. Okay.
Thank you so much for the talk. I was wondering if you've ever had miscommunication from the farmers of whether you're expanding this into genetically modified rooibos. Like, has that been a fear from the farmers? Or?
Yes, absolutely. It's the first question. Because they have heard about GMOs, but I don't plan to change rooibos. So the benefit is that if we find, and that is one of the tasks, to identify the genes that are, for example, associated with production of PPAG. PPAG by itself is not the only agent that can cure diabetes. So in rooibos, more and more research shows that it is the combination of different compounds that work together. So I'm never going to compete with the rooibos farmers and take the tea away. I'm only going to give them additional scientific proof of how healthy it is, that is, how useful it is. And in my dreams, we are going to actually start doing biotechnology in the Cederberg Mountains so that the people that are currently working on the fields, their children can start maybe even doing the lab work because they are connected to that plant. In my dearest hopes are to work with, so I don't know much about the medicinal plant in South Africa. I'm nearly done. I don't know much about the medicinal plants of South Africa. What I want to do is to collaborate with the different teams and establish working relationships with them, make sure that we have the paperwork that ensures benefit sharing, identify the people that can work with this and get their support, because the trust it's difficult to get them to trust you. We have one community where it's like, no, the doors are closed. Does this answer your question? Thank you.
Morobetsi from the South African Weather Service. So I'm just presenting on, so we have this project, we presented on it last year. So this is just a progress. So we've been conducting a number of experiments on the model development, and this is just another experiment we conducted recently, or in the past year. So just a background, numerical weather models So basically, we have physical processes in the atmosphere like rain, convection, and all that. So for us to predict them or how they're going to evolve in the atmosphere, we divide the globe or the area that we want to focus on into a number of grid points, and then we use the equations of physics with different assumptions to predict how they will evolve in the future. So basically, we write programs to describe those physical processes that we've used for the model, and then we run the model on an HPC, and in this case, we've used CHPC. So the need for numerical weather modeling or for model development is because did I press something? Okay, it's on again. Thanks. So at the South African Weather Service, we are the voice of weather for South Africa and the SADC. So we have to make sure that we produce weather forecasts and climate data and forecasts that are accurate and they are timely. And then for that to happen, we use models. So yeah, we need the models to be good for us to give warnings on time. And the other reason is because of climate change. I think we all know it's everywhere. We can even see how extreme events are just all over. Like one day you have a lot of rain, and another day it's like we've been having extended heat waves that I don't think we've had. This kind of weather as much as it is in the past. So also the projections for climate change show that we're going to have more droughts, extreme heat events, and more flash flooding events, and things like sea level rise, and that will result in things like increase in nutrition and infectious diseases, and food and water security risk, and as well as human health and safety risk. So, so we have the Paris Agreement that our government has signed under the UN SDGs. So part of it is that South Africa has committed to develop programs that will the plans and systems for adaptation to climate change, since now the focus is no longer on combating the impacts of climate change. So that should reduce vulnerability, build adaptive capacity, and build resilience to climate change on vulnerable communities. So for South Africa, the Paris Agreement that they signed, it's part of it says they're going to run different projects to help combat the impacts of climate change, including preparing for impacts of health as a result of extreme weather events. And that includes developing weather forecast models, which is what we are doing. Because basically models have been developed in first world countries, and so we just take them as they are and we run them. We don't know if it's going to capture our weather events well or in time. Though it works for them, it might not work for us. So we have to make sure that it works for us as well. So the South African Weather Service works with the CHPC. We have a number of applications that we run there. So this includes some running some numerical weather models for projects and research. So, so this one that I'm talking about is funded by the WRC, And yeah, there was a number of objectives in it, but this one that I'm presenting on focuses on the cumulus scheme, which basically is the part of the code that talks to how the model predicts rainfall and the other parameters that affect rainfall. So we use the CCAM from Australia, Cyro. So it's a seamless model, so it means we can run it for nowcasting timescales, which is up to 6 hours, 0 to 6 hours, up to maybe climate scales, which is decades. So yeah, we can use it for a number of applications, and it's also a stretch grid. So you can see that, okay, you can see, so this is the whole globe, right? And then for that area, okay, so it could be 25 kilometers on the outer domain. So, and then for this inner domain, maybe they could have chosen maybe to run it at 4 kilometers, and then for this one, maybe at 10 kilometers. So you can actually stretch it and then focus, make it high resolution for the area that you're interested in. Yeah, and then it also has a number of cumulus schemes. So, parameterization schemes, which includes a cumulus scheme. So the parameterization scheme is basically some assumptions that you make to address certain limitations within a model. So like the cumulus scheme, it addresses how the model predicts convection or precipitation. So this here is just, this here is just a part of the code that talks to how the model predicts precipitation at different grid scales. So if you run a model at 25 kilometers, how is it gonna produce rainfall? And then if maybe you move to 10 kilometers or 1 kilometer, it's all gonna be different, and that's what I'm gonna be talking about today. So this is a part of the SAGE-PC that, yeah, I just want to say thank you for, because I don't think we would have been able to do so much work without the SAGE-PC. So for this exercise, we had a number of case studies, and then we ran the model at 25 kilometers, 10 kilometers, 6 kilometers, 3 kilometers, and 1 kilometer. So those are the number of, we had a number of, different number of grid points for each run. So basically we would have like maybe 3 nests per run. So 25 kilometers was always there. So yeah. So it would be probably be 25 and then 10 and 3, and then 25, 6 and 1. Yeah, something like that. And then those are the number of grid points, and that's the size of data. It's quite massive. Yeah. And then another thing that we did differently from the previous ones, because we only have 600 nodes. So yeah, it was taking forever to get the models to run. So we reduced it so that we only run it over the area of interest, which worked in our favor in terms of completing runs quickly and saving space. But the limitation with it is if we want to actually diagnose the limitations within a model, the first thing we have to check is the mean sea level pressure patterns, which we can't check over a small domain. We have to have a larger domain for us to check that. But for now, I think it worked. I think, so we had 600 nodes, as I mentioned. The output files had 35 vertical levels, and we ran it for 48 forecast hours. And then the number of outputs, so we wrote it to write all the variables that were within the model because, we were not sure which one would have significance in terms of our research. So we just wrote all, and then we're going to investigate on that. And then the model, when the model runs, it produces raw output files which we post-process. So it gives us this, okay. Where is it? Okay. Well, the top ones, the ones that I wrote on the table, is the post-processed ones. So the 92 gigs is the raw data that we can post-process afterwards. And then this is just an example of the domains that we use. So the first one for the 1 kilometer. I'm sorry, for the 1 kilometer, so it looks something like that. It looks similar to the figure that I showed before. So you can see, like this whole area, it's 1 kilometer, and then only on the corner, that's where it's hanging with maybe pegs or something. And then the outside would be like 25 kilometers. And similarly to the 3 and 25 kilometers. Okay, so I'm just showing a few results of the simulations that we did. So here is the 1 kilometer run. Okay, let me start with the observations. So these are the ground observations. So we have a network of observations around the country. It's probably over 1,000. So this is 24-hour total rainfall. And then this is the IMEC data. And then it's satellite observations. And also this is satellite observation. It's Era 5 land, yes. So you can see this event, it was severe over there. It's like south of Gauteng. That's where, okay, for this one we had a tornado. So yeah, the rainfall was quite a lot over there, over Soweto. So if you check the model runs, the 1 kilometer, it almost located, well, it located that severe rainfall there in Soweto. And then the 3 kilometers missed it by just a bit. But when you go to the coastal resolutions, you can see that they didn't even pick up that event, but they actually had a larger spatial coverage, while the high resolution runs had a small spatial coverage. Similar to this case. So in this case, it's a case where we had some damage, severe damage in Twando. So it's over there, north of Limpopo. You can see that the models, yeah, they only captured the other rainfall south of Limpopo, but they didn't capture that one on the north where there was damage. And then, yeah, I think the pattern is similar with the other runs. This one is the case of Sun City when there was damage in 2018. So there's Sun City over there. So we can see on the observations that the observations captured it. It shows that there was a lot of rainfall. Though the Era 5 land didn't capture that, it misplaced it. But yeah, the observations and the IMEC data, they showed that. But the models were a bit slow. As you can see, for the 1 kilometer, it's a bit behind. And also for this one, it's also behind. They couldn't reach the area where the severe damage was. And yeah, you can see when we go to the coastal resolutions, we don't really have much rainfall. Then when we do, so for those areas that we displayed the rainfall, we took an area average, hourly average, and then we simulated, we plotted it. So we can see that for that, an area, okay, I'm trying to put it in a simple way. So the average of that whole area, so it shows that on average, the rainfall initiated by the model was much earlier than the actual observed. The observed also had a peak, which was slightly earlier, but well, it was quite close, but it was also much lower than the observed. So, so the dotted I think this one is not very clear, but there are dotted lines here. So the dotted lines show the convective rainfall. So the total rainfall for the model is the large scale rainfall plus the convective rainfall. So we plotted the total rainfall and the convective rainfall too. So the convective rainfall comes from the cumulus scheme. So when we plot that, we saw that it reduces with increasing resolution. So at high resolution, you have much less convection, which is supposed to be like that, but it switched off much too soon by 1 kilometer, so that the total rainfall is very little at 1 kilometer. So for this one, also the convective rainfall was quite small. And then also for this one, it was like, it was just the model of dynamics that produced rainfall. So the cumulus scheme didn't really produce much rainfall. So for some few vertical profiles, we took some vertical profiles for, this one is for a case of that tornado in Soweto. So at 1 kilometer, we see the blue, it's the relative humidity. We see that there's a lot more, the atmosphere was a bit more humid for the higher resolution, the 1 kilometer and the 3 kilometers, especially in the later hours of the day and the upper atmosphere. And then the temperatures, there were very little differences in the temperature, the red lines. And then the blue, I'm going to show you the detailed one. It's more visible on the next one for the blue. And then for the wind, there's wind barbs here. So at 9, it's 21 Zulu. It shows somewhere in the mid-troposphere. It shows wind shear. So wind shear is used to check, to identify the possibility of a tornado, which happened there. But here, the model shows it later at 1 kilometer. So I think that one was quite something remarkable of the model to just even indicate, even though it was late, it was able to pick that up. And it actually left us with a question to say, okay, if we run the model at much higher resolution, like at sub-kilometer resolution, would it be able to capture that tornado much earlier? Okay, so this one, the blue, is the one I was talking about. It's the omega, it's the vertical velocity. So what it shows is that at high resolution, the vertical velocity is stronger, and then it reduces as the resolution increases. So yes, I think that's how it's supposed to be. So it just says to the model developers that, okay, this one is fine. And then with the liquid water flux, I honestly couldn't notice any relationship with changing resolution. So that one, I think it would need more analysis. And then some more parameters, the boundary layer height. So the boundary layer height is also used to identify the cloud base. So you can see that all the resolution, the model runs, they produce the same level of boundary layer height, except where there was a lot of activity. And then you see that the blue and the green, which are the 1 and 3 kilometers, they have slightly higher boundary layer height compared to the coarser resolution. And then this is the cloud base and cloud top. So when there was no activity in the atmosphere, everything was all the same until when there was the thunderstorm. Then you can see that the blue and green, which is 1 and 3 kilometers, they are close to each other, and the coastal resolutions are also close to each other. So whether higher or lower, they are always close to each other, even the same as in the total cloud fraction. And the maximum CAPE and average CAPE, same applies to the latent and sensible heat fluxes. And this one was a bit more complex. Yeah, and it was also a questionable variable as to whether it's significant or not. So some vertical profiles for total cloud fraction. So this says, okay, at the surface, this is how much cloud we had, and then going up to 10, I think it's 10 hectopascals. Yeah. Then you can see in the middle troposphere, you can see that the blue and green, they have some sort of, well, they are always on one side, and then while the purple and yellow, which are the 6 and 10 kilometers, they are also on another side. Yeah. So in summary, the spatial distribution of rainfall for the coastal resolution, so the coast ones is the 25, from 6, 10, and 25. So those ones, they produce more rainfall, like on the spatial maps, like they had a larger spatial coverage, but they could not pick up the severe rainfall events. And also the models generally initiated rainfall much earlier than the observed. So with this ones, it just indicates the areas that we can start focusing on, like which part of the code can we work on so that the rainfall, maybe I think for now it's to say which part of the model scale awareness, or the part that addresses how much rainfall the model produces at different scales. How can we change that part of the code so that we have more convection or more convective rainfall, and that it doesn't switch off by 1 kilometer, but instead maybe go to maybe 300 meters, which, yeah, some authors have said that it should be switched off by 300 meters. So the challenges, I'm not sure if we really had challenges, considering that we reduced the domain. So the runs completed at a reasonable time. But yeah, like I mentioned, the problem with the domain that we chose is we can't diagnose the root of the problem. Like we can't get to see whether the model is producing the correct mean sea level pressure patterns. So yeah, I think if we can have more resources, then we'll probably be able to run for a bigger domain. And then, yeah, we didn't really have challenges. We just, yeah, we'll be grateful for more resources. And then going forward, we are doing more work to develop the model so that we are able to address issues like this, like why is this severe rainfall system, why is it moving slowly? What can we do so that it moves faster and it reaches the point where it's supposed to be, and we are able to capture or to give warnings in time for that. Yeah, and I want to thank all these people and organizations, and thank you.